Hello, everyone. I'm glad to present my work done at the Rockefeller University in Charles Rice Lab. It's a collaboration with Dr. Bill Schneider and Max Cass. First, I want to introduce the RNA launch system. We have shown that in vitro transcribed RNA can be transfected into a cell, got reverse transcribed, converted into cccDNA, and make new proteins like surface HBs protein. Figure 2 shows that we could take advantage of this, use RNA launch to test different classes of drugs like capsid inhibitors and reverse transcriptase inhibitors. The experiments were done by CLIA in 96 well plates, and we are looking at IC50s. We show that RNA launch could be used to test both classes of inhibitors. In figure 3, we try to test drug-resistant mutations by doing experiments with either capsid inhibitor, BAE, or limuvudine in wild-type and drug-resistant mutations. We show that T109I is resistant to capsid inhibitor, and M204V is resistant to limuvudine, and this is consistent to the literature. Next, in figure 4, we try to test either whether we could enrich these resistant mutations, because if we do, we might try to test whether we could enrich these mutations, because if we do, we might be able to identify resistant mutations in an unbiased way. In the mixed experiment, we show that only M204V could be enriched, and that makes sense because polymerase has been predicted to work in cis, but core work in trans. Now I want to move to the part 2, the DMS RNA launch part. We have combined the RNA launch method with deep mutational scanning developed by Jesse Bloom's lab. Schematics shows that we could transfect the randomized library into cells, extracted DNA to sequence all the variants and compare with the input variants. What we're looking at is the heat map. The y-axis shows that there are 64 codons. X-axis means all the positions at the polymerase region. Red color means they are negatively selected, and the yellow color means they are neutral. If we zoom out, first we see that most of the stop codons on top of the figure are strongly negatively selected, meaning that the assay works really properly. Most regions at TP RNA and RNase-H are essential, but the spacer region is really neutral for the polymerase function. One thing I want to mention is that our assay really focuses on the polymerase region because only pol work in cis. That's why we peel off the selection by S open reading frame, which might give selections at the spacer region. Next, I want to move to the part 3. We want to test whether RNA launch method could be used to predict the drug-resistant mutations. At the figure 6, we show that by using regular RNA launch, we could identify M204V, M204I, two most observed clinical mutations. We think that these mutations are introduced by the era of T7 polymerase. At figure 7, we show that if we use the DMS RNA launch method, now we observe a lot more mutations. If we map DMS mutations into a 3D structure in the figure 8, we show that all these mutations are clustered together. Red ones are those observed in clinics, and pink ones are novel mutations. The question is why these pink ones are observed in clinics, with reason that maybe these pink ones are negatively selected from the open reading frame as a surface domain. An indirect improvement is that when we mapped the clinical observed mutations in HIV into its crystal structure, we found that these mutations resemble to the DMS HBV ones because HIV do not have surface protein as overlapping open reading frames. In summary, we show that RNA launch could be used to test the drugs and resist mutations. By DMS, we show that we acquired the fitness map in the whole polymerase region, and in part 3, we show that we could identify drug-resistant mutations by on a launch system. That's all, and thank you very much.